I am Larry Hetcher. This is Psych 302, Computer Applications and Data Analysis. And today is going to be part one of our coverage of lecture notes, chapter 125, ANOVA with two within subject factors. As always, a reminder to my Psych 302 students, you do not need to download the data set or perform the analyses described here. Simply use these lecture notes as a guide when you complete your exercise, which will involve a different data set. This chapter is divided across two class meetings. We'll cover pages 1 through 33 during our initial class meeting, that is today, and we'll cover pages 33 through 47 during the second class meeting. So this is part one. I'm on page 2. An introduction, ANOVA with two within subject factors basic concepts. This chapter covers another member of the family of procedures called analysis of variance, or ANOVA. Procedure covered here is two-way ANOVA within subjects design. The analysis performed on a single continuous criterion variable. The analysis involves two predictive variables, that is, two factors, independent variables if it's a true experiment. Generically, this chapter will refer to one of them as factor A and the other as factor B. Each predictor is typically a limited value variable, one that consists of 2 to 6 conditions or levels. We'll compute the mean score on the criterion variable for each of these conditions. Both predictive variables must be within subject factors, that is, repeated measures factors. These constructs are typically created by using a repeated measures procedure in which a given participant is exposed to every condition under the independent variable and provides a separate score on the criterion variable for each of these conditions. Let's make things concrete now. Illustrative investigation, screen resolution, screen size, and reading enjoyment. In a previous chapter, you conducted an investigation with a new line of e-readers that college students could use to read electronic textbooks, articles, and such. Corporation for which you did the work was pleased and subsequently asked you to conduct a similar investigation in which you manipulated two independent variables instead of just one. This new study would be based on a sample of 10 college students. Once again, the dependent variable will be reading enjoyment, ratings of how much the participants like using a specific e-reader. Scores could range from 1 to 100. Higher scores indicate greater self-reported reading enjoyment. Factor A, your first independent variable. With this revised study, one of the independent variables, that is factor A, was a dichotomous variable called screen resolution. It had two levels. When being exposed to the low resolution condition, participant used an e-reader with a screen that displayed relatively small number of pixels per inch, producing not terribly sharp letters and images. We proceed to page 3. We're still talking about factor A screen resolution and the second level. When being exposed to the high resolution condition, participant used an e-reader with a screen that displayed relatively large number of pixels per inch, producing sharper letters and images. So that is factor A, screen resolution. We come to factor B, screen size. With this revised study, the other independent variable, that is factor B, was screen size. Same limited value variable that you manipulated in chapter 120. This factor consisted of 3 levels. When being exposed to the 6-inch screen condition, the participant used an e-reader with a screen that measured 6 inches diagonally. When being exposed to the 10-inch screen condition, they used an e-reader with a 10-inch screen, and when being exposed to the 12-inch condition, the participant used an e-reader with a 12-inch screen. Figure 1 below illustrates the factorial design used in the current investigation. In the figure, the two horizontal rows represent the two conditions of factor A screen resolution, and the vertical columns of the figure represent the 3 treatment conditions of factor B screen size. So here, and where are we? Bottom page 3? Yes, bottom page 3, we have our factorial design. Factor A, screen resolution. This row represents the folks when they're in the high resolution condition, and this represents them when they're in the low resolution condition. Factor B, screen size. This column represents them in the 6-inch condition, in the 10-inch condition, and 12-inch condition. Notice within a given cell, I indicated all 10 participants. And that's because each of the 10 participants was exposed to both conditions under factor A and all three conditions under factor B. I continue to page 4. In this investigation, given participant used an e-reader with a low-resolution 6-inch screen for 2 weeks and provided a score on reading enjoyment dependent variable. Same participant then used an e-reader with a low-resolution 10-inch screen for another 2 weeks and provided a separate score on the same reading enjoyment dependent variable. Participant continued in this way for 2 weeks with each type of screen, and by the end of the 12-week investigation, this participant had provided 6 separate scores on the reading enjoyment dependent variable, one for each combination of screen resolution and screen size. Still on page 4, new section begins. Types of effects that are possible in a two-way ANOVA. Now this should sound familiar. In a previous chapter, you learned about two-way ANOVA with two between subject factors, and those basic concepts apply to two-way ANOVA with two within subject factors as well. In fact, that's how my section begins. In chapter 100, with two between subject factors, you learned that 3 types of effects are possible when one performs two-way ANOVA with two between factors. Same basic types of effects are possible when we perform ANOVA with two within subject factors. I should really stop speaking off the cuff because everything is in writing here, isn't it? Okay, this section will briefly illustrate each type of effect. You might get a main effect for factor A. When the main effect for a given factor in two-way ANOVA is significant, it indicates there's a difference between the means of at least 2 conditions under that factor. 2 main effects are possible with the current research design. The first of these is the main effect for factor A screen resolution. Following a research hypothesis predicts a main effect for this factor. Mean reading enjoyment scores obtained in the high resolution condition will be higher than those obtained in the low resolution condition. Following figure illustrates fictitious outcome that would provide support for the preceding research hypothesis. Here it is. If you got results like this, you would have a main effect for screen resolution. Screen resolution is represented by 2 lines. The solid line represents the scores that people provided when they're in the high resolution condition. The dashed line represents the scores they got when they were in the low resolution condition. If you obtain results like this, you can see that the reading enjoyment was higher in the high condition than in the low condition. That would indicate you've got a main effect for screen resolution. That's not the only main effect that you might obtain. You might get a main effect for factor B. Second type of main effect is possible in two-way ANOVA is a main effect for factor B. In the current study, factor B is screen size. This factor consisted of 3 conditions: 6-inch screen condition, 10-inch condition, 12-inch condition. If the main effect for factor B were statistically significant, it would indicate there's a statistically significant difference between at least 2 of the 3 conditions. The following research hypothesis predicts a main effect for this factor. It says mean reading enjoyment scores obtained in 12-inch condition will be higher than those obtained in the 10-inch condition, which in turn will be higher than those obtained in 6-inch condition. Following figure illustrates fictitious outcome that would provide support for this research hypothesis. And yes, sure enough, this would be a main effect for screen size. Notice the screen size factor is represented by 3 points on the horizontal axis. Here's the condition: 6-inch screen condition, 10-inch screen condition, 12-inch screen condition. If you got results like this, it would mean reading enjoyment scores were higher in the 12-inch condition than in the 10-inch condition, and they were higher in the 10-inch condition than in the 6-inch condition. That would indicate you've got a main effect for screen size. Now, main effects in factorial ANOVA is not much different from treatment effect in one-way ANOVA, but there's one kind of effect that you can investigate in factorial ANOVA that you can't investigate in one-way ANOVA, and that is this concept: interaction between factor A and factor B. In chapter 110, you learned that a significant two-way interaction indicates relationship between one predictor variable and the criterion variable is different at different levels of the second predictor variable. For example, imagine you reviewed previous research on e-readers. You'll learn that when people are using high-resolution screen, they don't care much about the size of the screen. They're happy with high-resolution screens that their reading, they are so happy with high resolution screens that their reading enjoyment scores are high, regardless of whether they're using a 6-inch screen, 10-inch screen, or 12-inch screen. However, previous research suggests that a different relationship when people use a low resolution e-reader. When using a low resolution e-reader, they find a 6-inch screen is too small. They're happier with a 10-inch screen and happier still with a 12-inch screen. Review of the previous research led you to state the following research hypothesis. There will be an interaction between screen size and screen resolution, such that there'll be a relatively strong positive relationship between screen size and reading enjoyment when using low resolution screen, but there'll be no relationship between screen size and reading enjoyment when using a high resolution screen. Concept of interaction is easiest to understand when illustrated in a graph. The following figure therefore illustrates the nature of this predicted interaction. On page 7, I'm at the top of page 7. We have a graph that illustrates an interaction between screen size and screen resolution. Notice that when you focus just on the people that have a low resolution screen, that's the dashed line. If they're using a low resolution screen, then the bigger the screen, the happier they are with it. Maybe a better way to say it is if you're using a low resolution screen, the smaller the screen, the more unhappy you are with it. On the other hand, if you focus on just the solid line, that represents the people when they're using a high resolution screen. Size doesn't really matter much. They are equally happy with a 6-inch screen, 10-inch screen, and 12-inch screen. The concept of interaction, let's leave this figure showing. Concept of interaction is all about it depends. Previous figure shows that the nature of the relationship between screen size and reading enjoyment depends on whether the participant is using low resolution screen versus a high resolution screen. When illustrating results using a line graph such as the one presented above, hallmark of a significant interaction effect is non-parallel lines. If one of the lines in the figure is not parallel with another, it often indicates you've got an interaction. And sure enough, notice this line is not parallel with this line. And simply eyeballing it like this is not enough to know we've got a significant interaction. To determine that, we will need to do the statistical analyses, which of course we will. I'm on page 8 now, halfway down page 8. This chapter will focus on interaction effects, not main effects. This chapter will not focus on the interpretation of main effects. In part, this is because main effects were covered in the previous chapter. Lecture notes on chapter 100, ANOVA with 2 between subject factors, main effects only. So you learn how to make sense of main effects in that chapter. In this chapter, we're going to focus on a totally different kind of effect, and that is the interaction effect. So let's get started with the program. Now again, you folks are not doing this, you're just using this as a guide. Starting Jamovi and opening the data set. Let's continue to page 9. I'm near the top of page 9. Downloading the data set. Before you perform any of these tasks, be sure you've already gone in Canvas modules, located the data file, downloaded it onto the drive where you plan to save your Jamovi data sets and output. Here's the name of the file that I happen to be using with this illustrative chapter. Starting the Jamovi application. As you know, the way you start Jamovi depends on whether you're using the installed version or the online version. And in that section, I give you a reminder of how it's done. Let's continue to page 10. I'm at the top of page 10, and we have a red font box. What does it say? For the upcoming exercise, Hatcher's 302 class will download an existing data set. And that should be a relief to you folks. You don't have to type the data set from scratch. You'll simply download a data set that I make available in Canvas. Finding and opening your Jamovi data set. Now that Jamovi is open, and we assume you've opened Jamovi, you'll use the Jamovi file panel to locate the data file and open it into the Jamovi spreadsheet. So this is what your initial Jamovi interface looks like when you start it. At the top left of the Jamovi interface, select the file. That is the air vent icon. These 3 horizontal lines. and click it. In the resulting file panel, this is the file panel, in the resulting file panel, select Open. In the resulting Open panel, select Browse. And in the resulting Open dialog box, navigate your way to the drive where you had saved your Jamovi data file, double-click the name of that data file, and the data file opens in the Jamovi spreadsheet. You're now ready to perform the analyses. I have a note here. If anybody is watching this video and they don't have a data set that some kindly professor made available to them, they have to key it themselves. Be aware that if you need guidance in creating your own Jamovi data set, see the chapter appendix, Creating the Current Data Set from Scratch, which appears at the end of this chapter. Let's continue. What page was I on? I'm on page 11. Page 11, we go to page 12. This is the data set, the way that it would look in table form. In a minute, we're going to see the same data set in the Jamovi spreadsheets. We'll hold off and focus on it when we get there. Okay, now I'm on page 13 and about halfway down page 13. The Jamovi data spreadsheet. Once you've downloaded the Jamovi file needed for the current analysis, it should resemble the following screen capture. Your Jamovi dataset is going to have 7 variables. Now, the first one is headed participant. It's merely assigns a unique participant number to each of the 10 participants, and notice that there are only 10. I had created a variable that I called low res 6-inch, and we have numbers in that variable. What is this? These are the reading enjoyment scores that fictitious participants displayed when they were using a low-resolution 6-inch screen. We have different set of numbers in this variable. It is headed low-res 10-inch. These are the reading enjoyment scores they displayed when they're using a low-resolution 10-inch screen, and so forth for every possible combination of conditions. The last variable is headed high-resolution 12-inch, and this is the reading enjoyment scores they displayed when they're using a high-resolution screen with 12-inch diagonal length. So there's our Jamovi spreadsheet that has the data set that we're going to analyze. Now remember the names of these variables because they're going to appear in the Jamovi program that we are about to put together. I'm on page 14 now. Put in the black and white the things I just said. I'm going to skip this because I've already said it. What's in yellow font here is what I just said off the cuff, which I said I was going to stop doing. I'm on page 14 now, halfway down page 14. Now, let's request some statistics, requesting plots and descriptive statistics. In part, we do this so that we can see whether we meet the assumptions underlying 2A ANOVA with 2 within subject factors. In our Jamovi interface, select the Analyses tab at the top, if necessary, to reveal the Analyses ribbon. There, select the Exploration icon, which is this one. It produces a drop-down menu, and there, select Descriptives. This procedure produces the initial analysis options panel for the Descriptives procedure, as shown below. Below the heading Descriptives in the variable list box, the box displays the names of the variables in the data set. And sure enough, this is the analysis options panel for the Descriptives procedure. And in the big box on the left, we see the names of all the variables that were included in that Jamovi spreadsheet I showed you earlier. Low-res 6-inch, low-res 10-inch, and so forth. You will now modify the Descriptives analysis options panel so that it looks like the one in the figure below. And I give you a list of all the things you're going to do. I'm on page 16 now. Move the name of the quantitative criterion variables here. We did have the 6 variables here that contain reading enjoyment scores obtained under the various conditions. Take those variables and move them over to the box on the right headed variables. Then check these boxes in the statistics section so as to order the statistics and the options we desire. The students in my class, you should look very closely at this and make sure that you check and uncheck exactly the same boxes that I have here. And in the plot section, check these options to select the plots and plot options that we want. If you're a student in my class, you need to do an additional thing. Add your name to the results panel so that I know you're handing in your own program and you're not handing in your roommate's program. We'll go to page 17. Top of page 17. Click the heading results and then type your name here so that your name will appear in the results section. So we have requested some descriptive statistics, checking assumptions, and screening data. Statistical assumptions refer to the characteristics your data must display for the data analysis procedure to produce correct results. We have the usual long section here in which I talk about various descriptive statistics you should review to verify that you've satisfied assumptions. To refresh your memory, now this is what a descriptives table looks like that's produced by Jamovi. On the left side, we have the names of the various statistics, such as mean, median, and so forth. These columns provide statistics for the criterion variable, that is reading enjoyment, obtained under each combination of conditions. When students were using the low resolution 6-inch screen, what was the mean score they displayed? 36.10. What about when they're using the low resolution 10-inch screen? The mean was 50.50 and so forth. Do things look okay so far? Well, first row is headed in, and yes, it looks like all 10 participants gave us usable data. That's good. Means and standard deviations appear in these rows. We'll take a closer look at them later to see if they're giving us the trend that we had predicted. In seeing whether there's any obvious problems with the data set, no minimum or maximum values are out of bounds. Remember, our reading enjoyment scores could not be any lower than 1 or any higher than 100. So if we saw a negative number in minimum, that would be bad news. And if we saw a value in maximum that was bigger than 100, we would know we made some kind of error in keying our data. No problems like that. And I have a note here, no serious problems with skewness or kurtosis. Here we have skewness statistics and kurtosis statistics explained in earlier chapters, and I'll make a quick mention of the skewness statistic here. Let's go to page 19. To refresh your memory, and I'm doing this just to refresh your memory on one of the stats that we look at to know if our data satisfies some of the basic assumptions of a procedure. On page, what did I say it was, 19, middle of page 19. The skewness statistic indicates the extent to which distribution is skewed, with one tail being longer than the other. Huck points out that many researchers view a distribution as being approximately normal if the skewness statistic is somewhere between -1 through +1. The descriptives table that we looked at above shows that the skewness statistic was within these limits for each of the 6 subconditions. Combined, these results indicate that none of the 6 subconditions displayed a substantial skew. What is he talking about? To the right of the heading skewness, is there anything smaller than -1.0 or bigger than +1.0? That's okay, that's okay, that's okay. Okay, all these skewness statistics were between -1 and +1. Doesn't mean our data is perfect, but it does indicate there's no apparent problem involving skewness. And I give similar instructions on evaluating other characteristics of the variables. We'll scroll through the QQ plots. You already know how to interpret those. We go to page 20, the bottom of page 20. Conclusion regarding assumptions. Results reviewed so far are consistent with the idea that reading enjoyment scores display an approximately normal distribution under each of the 6 combinations treatment conditions. So far, so good. There could still be problems with our data, but no obvious problems so far. We come to page 21 and a section headed Requesting the ANOVA. If you're a student in my Psych 302 class, you remember me saying, using Jamovi is not intellectually difficult, but it is tedious in that it involves many steps. And no procedure involves more steps than analysis of variance with 2 within subject factors. You're going to find the things we're about to do are not intellectually challenging, but there are many steps that we need to follow in order to request the ANOVA. So be prepared to take a close look at these lecture notes. And when you're actually analyzing your data set, make sure you replicate my steps using appropriate names that are relevant to the data set that you're analyzing. So, requesting the ANOVA. To get things started, select the Analyses tab if necessary to reveal the Analyses ribbon. There, select the ANOVA icon. It'll produce a drop-down menu, and there you'll select Repeated Measures ANOVA. Go to page 22. We are looking at the Repeated Measures ANOVA Analysis Options panel. We have the usual variable list on the left side. The large box on the left contains the names of the variables that already existed in the data set that you downloaded. Now we have a number of boxes on the right. Some of these we can ignore, but a couple of them we can't ignore. For example, there is a box headed Repeated Measures Factors, the Repeated Measures Factors box. In this box, we will tell Jamovi the names of our factors. What name did I give to my first independent variable? What name did I give to my second independent variable? And furthermore, we will tell Jamovi what name I want to give to level 1 under my first factor, what name I want to give to level 2 under my first factor, and so forth. Let's take this step by step. I'm on page 23 now. To get started, click this label and replace it with resolution. Remember, the names of one of our factors is resolution, as in screen resolution. This heading that is in bold begins by being named RM factor. That means repeated measures factor 1. Now we could just leave it like that, but when we look at the output, it'll say RM factor 1 in our output, and we're not going to remember what RM factor 1 was. So we're going to replace this with a meaningful name for our factor. I'm going to click that and replace it with the word resolution. So we'll know that's the screen resolution factor. This is the name of one of the levels, one of the conditions under the factor. It starts off being level 1, but we want to give it a meaningful name. The two factors under resolution was low resolution and high resolution. So click that and replace it with the name low. Click the second one, level 2, and replace it with the name high. So we now have name of the factor is resolution. One of the levels is named low. One of the levels is named high. So we've taken care of one of our factors. On page 23, we show what the repeated measures factors box should look like at this point. Name of one of our factors is resolution, and here's the names of the two conditions, low versus high. But we're not done. We have a second factor, don't we? Next task is to click this label and replace it with screen size. Then scroll down to reveal the empty level labels below. RM factor 2, repeated measures factor 2. Click this and replace it with screen size. I go to page 24. Okay, I've clicked it. I've replaced that name with screen size. Here, it initially gives the levels names like level A, level B. Click level A and replace it with the name 6-inch for 6-inch screen. Click level B, replace it with 10-inch screen. Click level C, replace it with 12-inch, as in 12-inch screen. When finished, the repeated measures factors box has now been completed. The resolution factor has been scrolled up. You can't see it anymore, but it's up above. And our second factor is the screen size factor. We've given meaningful names to the levels under screen size. Okay, so we finished repeated measures factor box, but we're not close to being done. Next, we need to take care of the repeated measures cells box. Now, notice that Jamovi has already created names on the right side. It says low 6-inch, low 10-inch, low 12-inch. Jamovi is telling us that one of the cells in our research design contains scores that participants provided when they're in the low resolution condition with a 6-inch screen. One of the variables contains enjoyment scores that the students provided when they're in the low resolution 10-inch screen. Jamovi is asking us which of your variables contains the enjoyment scores obtained under the low 6-inch screen? Which of your initial variables contains enjoyment scores obtained under the low 10-inch screen? All we have to do is tell Jamovi which variable contains those scores. So we start that process on page 26. Initial list of variables is in the big box on the left side. Select the variable named low res 6-inch. And that's the name I gave to the variable when I created it. Before you downloaded the data set, I'd created a data set that had 6 data variables in it, and one of them I gave the name low res 6-inch. Select that and move it over to this row. Move it over to the row that Jamovi had already labeled low 6-inch. Do the same with all 6 of those variables. Let's scroll back to page 26 to make this clear. My variable low res 6-inch, drag it over here so that Jamovi knows that's the variable that contains the scores that students provided when they're in the low resolution 6-inch screen. Grab low res 10-inch and move it over to this row so that Jamovi knows that's the reading enjoyment scores that people provided when they're in the low resolution 10-inch screen, and so forth. So you move each of my quantitative variables here, put them in the correct row here. Continue in this fashion until you've moved all 6 data variables appearing in the variable list on the left side of the interface into the appropriate row of repeated measure cells box on the right. When finished, your analysis options panel should resemble the following. And you can't see the very top row because I scrolled up some, but if you go to the next row, it's variable low res 6-inch. That's where you'll find enjoyment scores for the low resolution, or rather low res 12-inch. That's where you'll find enjoyment scores for the low resolution 12-inch screen. The variable called high res 6-inch contains scores for the high resolution 6-inch screen, and so forth. Repeated measures cells box is finished. But no, we are not finished. There is more to do. I'm on page 28 now. Top of page 28, we're going to need an omnibus index of effect size. So check the box headed partial eta squared. We use that as our omnibus index of effect size. Tells how big the effect is for the main effect for factor A, how big is the main effect for factor B, and so forth. Under the heading dependent variable label, you could just leave this as it is, but it's better to replace it with a meaningful name for your dependent variable. And remember, our dependent variable was reading enjoyment. So why don't we click this box and type enjoyment there? That's the name of our dependent variable. I am still on page 28. We come to the section requesting tests of sphericity. Sphericity is one of the assumptions that ideally should be satisfied if you're going to do a within subjects ANOVA. This is where we will request it. If necessary, click this arrow to reveal the assumption checks panel. Click this arrow to reveal the section of the panel, and there check sphericity and check none. You're telling Jamovi, give me the results of the sphericity test so I know whether I've satisfied that assumption. And when it comes to sphericity corrections, for the moment, we'll check none. If for some reason we want greenhouse geyser correction or the Hun-Felt correction, we could check these boxes, but we are not going to need those, as we shall see later. Let's continue. I'm on page 29, requesting post hoc tests. One of our independent variables, screen size, has 3 conditions. And anytime you have an independent variable with 3 or more conditions, you probably should request post hoc tests so you can see which conditions are significantly different from one another. Now, you'll ignore those results if your factor is involved in an interaction. But at this point, you don't know whether it's going to be involved in an interaction or not, so you might as well go ahead and request it. There is a bar headed post hoc test. Click the arrow to reveal its contents if necessary. Here's what we're going to do. I'm on page 30 now. I only have one factor, one independent variable that has 3 or more conditions, and that is screen size. So select screen size and move it to the large box on the right over here. Uncheck Tukey and check Bonferroni so that you're requesting the Bonferroni post hoc test. When finished, this is what your post hoc test panel should look like. Give me post hoc test for the factor called screen size, and the specific kind of post hoc test I want is the Bonferroni post hoc test. And we are still not finished. If we did these analyses and things came out statistically significant, we wouldn't necessarily know which conditions displayed the highest mean scores. We've got to tell Jamovi to provide us with those condition means. And we will do that beginning at the bottom of page 30, requesting tables and plots of means. On page 31, we have a bar that's headed estimated marginal means. If necessary, click the arrow to reveal the contents of this estimated marginal means panel. Move the variable resolution to this term 1 box, and then click add new term and move screen size to the resulting term 2 box. What are you saying? Under the heading marginal means, we have this heading term 1. Jamovi is asking us, do you want me to report mean scores broken down by one of your factors? And the answer is, yeah, I want mean scores broken down by resolution. So I select resolution and I put it there. But wait a minute, there's another factor that I want to see mean scores for, and that's my screen size factor. There's no place to put screen size, so I have to click this blue box where it says add a new term, and it will create a new empty box for me. Yep, when I clicked that blue box, it created a new empty box called term 2. So I selected screen size and I moved screen size there. There's one last way that I need to have my means broken down. If my interaction effect is significant, I need to have the means broken down by resolution and screen size simultaneously. That is, I need to see the mean scores for each of the 6 cells of my research design. So I'm going to have to request that. To get the ball rolling, I click this blue box that's headed Add New Term. Proceeding to page 33. So I click the blue box and it created a new empty box for me that initially was headed Term 3. I will move the variables screen size and resolution into this empty box. And because both screen size and resolution both appear in the empty box, that tells Jamovi, I want you to give me means broken down by both of those factors simultaneously. In other words, give me the mean score displayed by each of the 6 cells of my research design. So I'm going to have mean scores broken down by resolution, broken down by screen size, and also broken down by screen size and resolution simultaneously. We do want to request options, so check all the boxes indicated here so that we get tables of means, plots of means, confidence intervals around our means. So check all those boxes to request all options. And finally, enough already. We have now requested ANOVA with 2 within subject factors, test of sphericity, post hoc tests, tables of means, and plots of means. That is plenty for one class meeting. So I have the red font box here that says, if you're a student of mine, begin work on Jamovi Exercise 125.01 ANOVA, 2 within factors. For today, you'll complete only task 1, performing the analysis in Jamovi. You will complete task 2 and task 3 during next week's class meeting. You do not need to upload anything today. You do not need to complete any multiple choice quiz today. You will do that next week. For today, you're going to have your hands full just making all the requests that I have just described so that you can do your ANOVA. So this is part 1 of lecture notes, chapter 125, ANOVA with 2 within subject factors.